Now let's talk about the general factors which are required for the regulation of the erythropoiesis. Now these general factors which are required can be first of all hypoxia or the reduced amount of oxygen being delivered to the tissue. Now this stage usually contributes to the formation of RBCs. Hypoxia can induce increased amount of erythropoiesis at the erythropoietic sites. Another general factor is the recovery from anemia. Whenever a patient is recovering from anemia, now anemia is a condition where a patient suffers from low amounts of hemoglobin in the body. Now, whenever a patient is recovering from any type of anemia, that the hemoglobin is starting to recover and the amount of RBCs has started to rise, this can in effect affect the regulation of erythropoiesis and can induce increased amount of formation of the RBCs. Another one is the ineffective erythropoiesis. Now, ineffective erythropoiesis due to a variety of factors, either due to the lack of the maturation factors or the lack of the bone marrow, or there's some kind of immunosuppressive element or myelosuppressive element is present there. Whatever the case is, if there is ineffective erythropoiesis, it will signal other sites of erythropoiesis to make more of RBCs. So if one site is compromised, the other sites are signaled to up their formation of RBCs. Another one is the hemolytic anemia. Now, hemolytic anemia is the one where both the number of the RBCs is reduced and the amount of the hemoglobin circulating in the blood is reduced. So both the number of the RBCs and hemoglobin both are reduced in hemolytic anemia. And hemolytic anemia is due to the hemolysis, that is the destruction of the RBCs in the blood. And because of this, the number and hemoglobin totally plummets. And because of this, whenever a patient is suffering from the hemolytic anemia, erythropoiesis and erythropoietic sites are regulated to increase their amount of regulation and the amount of RBCs being produced and then being placed into the circulation. Last but not least is the blood loss. Whenever in the body there is some evidence of blood loss or hemorrhage, it signals the erythropoietin and other factors to increase the amount of erythropoiesis. In fact, hemorrhage or blood loss is considered one of the primary cause of the release of erythropoietin. And with erythropoietin, erythropoiesis increases. So all of these regulate the amount of erythropoiesis in erythropoietic sites due to the increased amount of the erythropoietin. Now, erythropoietin is a glycoprotein. It is a special glycoprotein which is released by certain cells which are found inside the kidney. There are certain differentiated fibroblasts which are present in the kidneys which release the erythropoietin. Now, once this glycoprotein or erythropoietin is released, it increases the amount of the formation of RBCs at the erythropoietic sites. So, let's talk about erythropoietin, because it is due to erythropoietin that erythropoiesis is regulated. If there is increased amount of erythropoietin, there is increased amount of erythropoiesis. If there is low amount of erythropoietin, erythropoiesis is decreased. So erythropoietin is a glycoprotein, like I said. Now this glycoprotein contains 4 chains of oligosaccharides and 4 chains of the amino acids. Now this glycoprotein has a variety of effects. It does not only regulate the erythropoiesis, it has certain other effects too. The first one and the primary one is the effect on the erythropoiesis. Now it does so by increasing and affecting the amount of the progenitor cells which are present inside the bone marrow. Another function of erythropoietin is the protective function. Now, it protects the brain and the hypoxic states of the body from the brain by increasing the amount of oxygen being delivered to the brain. So, if the body is suffering from hypoxic state, the brain will suffer from hypoxia at a later stage. It will not immediately have the effects of hypoxia, and it is due to the protective hypoxia preconditioning of the erythropoietin. And because of this, variety of other metabolic functions inside the brain are also affected. One of those is the appetite. Another function of erythropoietin is its effect on the white fat. Now, what happens is that it increases the amount of white fat which is normally present inside our body. Now, during the puberty, the amount of the fat which is present inside the human body increases, and it increases at a certain site. Now, the amount by which the white blood, the adipose tissue is increased, and the sites where it increases is regulated by the erythropoietin. Now, it does so by one having a protective effect on the white cell, white fat. Now, because of the white fat, there's increased amount of oxygen consumption. The mitochondria increases again because the oxygen consumption is increased, and because of this, we have an anti-inflammatory effect. So, erythropoietin increases the amount of white fat which has a protective function. And because of this, because of increased amount of white fat, we have increased amount of oxygen consumption. So, because of the effects on white fat, it also increases the amount of the erythropoiesis because there is huge demand of oxygen, and that has to be met by the increased number of the RBCs which are being produced. Another effect of erythropoietin is the protective effect on the skeletal muscles. Now, it does so by the amount of the myoglobin that is present inside the skeletal muscles. More the amount of the myoglobin and the amount of oxygen that that myoglobin contains is the amount of protection that skeletal muscle will have. So it has a protective effect on the skeletal muscles. Another function of it is that it has a protective effect on the heart. Now, its protective effect on the heart is on the endothelium. It is not primarily on the myothelium of the heart, it is on the endothelium of the heart. And it does so by maintaining the vascular tone of all the coronary arteries which are present in the endothelium of the heart. Now, when the erythropoietin maintains the vascular tone of all the coronary arteries, arterioles, it maintains the flow of the blood around the heart and to the tissue of the heart. And that gives the heart the protective function through the erythropoietin. Now, because of all of this, whenever erythropoietin is released by the kidneys, what happens is there is increased amount of one erythroferrone and erythropoiesis. Now, erythroferrone is a byproduct of the erythropoietin formation and is usually found circulating in the blood. So increased amount of erythroferrone will signal increased amount of the erythropoiesis being taken place. Now this will give further rise to the increased amount of the hepcidin which is being produced by the liver. Now when the liver produces least amount of hepcidin, what happens is that two of the things happen. Now, first thing is that it increases the amount of the iron reabsorption in the gut. So, what happens is that when we eat any iron containing vegetable or meal, iron is not usually absorbed. It is only absorbed when there's either some kind of iron transporter that is present there, which takes the iron from the lumen of the intestines into the bloodstream. The second effect of low amounts of hepcidin is that it increases the amount of iron being released from the iron stores. So all the cells which store the iron in them will start to release iron into the bloodstream. So, in effect, erythropoietin has two effects. One, it will increase the absorption of the iron from the diet into the bloodstream. Second, all the iron deposits found inside the body will start to release the iron into the bloodstream. Now, whenever this happens, like I said, it happens in the gut because of some kind of transporter, and that transporter is known as the DMT1. And that DMT1 is the transporter or the intestinal ferroportin, which increases the amount of iron being transported from the lumen to the bloodstream. And whenever there is increased amount of erythropoietin, there is increased amount of expression of this transporter in the lumen of the intestines. So whenever there is increased amount of iron absorption, what happens is there's increased amount of iron availability for erythropoiesis, because like we know that hemoglobin has three main components: the heme part, the globin part, and the iron which is found inside, trapped inside the heme part. So hemoglobin synthesis cannot take place by first having without having the iron. So iron has to be present first of all before either the heme synthesis or the globin synthesis can take place. So because of erythropoietin, there is now increased amount of iron availability for erythropoiesis or for the formation of the hemoglobin that is found inside the RBCs. So when there is increased availability for erythropoiesis, this in turn increases the iron release. So it contains a kind of a loop feedback mechanism. Least amount of hepcidin will increase the amount of absorption, will increase the amount of iron in the blood. Now increase amount of iron in the blood will store it. Now because of the storage and decreased amount of hepcidin, the iron will start to release from it. So because of both of these function, erythropoietin has huge effect on the formation of the RBCs.